Hello, good morning everyone. Welcome to my channel. Today my topic is enzymes involved in gene cloning. First of all, let us learn about what is cloning. Cloning is the process of producing similar process of genetically identical individuals that occur in nature. Cloning also refers to process used to create copies of DNA fragments. Gene cloning. Gene cloning is the act of making copies of a single gene. Cloning can provide a pure sample of an individual gene, separated all the other genes that it normally shares the cells with. Once a gene is identified, clones can be used in many areas like industrial biotechnology, biomedical research, medical biotechnology, and to provide vaccines, various other branches. Enzymes involved in gene cloning: nuclease, polymerase, ligase, transferase, DNase, RNase, kinase, phosphatase. First we are going to learn about nuclease. A nuclease is an enzyme capable of cleaving the phosphodiester bonds between the nucleotide subunits of nucleic acids. Exonucleases are enzymes that work by cleaving nucleotides one at a time from the end of a polynucleotide chain. A hydrolyzing reaction that breaks phosphodiester bonds at either 3' or 5' end occurs. Nucleases are mostly classified into two types based on its function, that are exonuclease and endonuclease. Exonuclease. It cuts phosphodiester bonds towards ends. It does not cleave DNA strands with terminal 3' OH groups because they are blocked by phosphoryl or acetyl groups. Endonuclease. It cuts the phosphodiester bond within a polynucleotide chain, in contrast to exonuclease, which cleave phosphodiester bonds at the end of a polynucleotide chain. Polymerase. A DNA polymerase is an enzyme that catalyzes the polymerization of deoxyribonucleotides into a DNA strand. This process copies a piece of DNA. The newly polymerized molecule is complementary to the template's original partner strand. DNA polymerase uses magnesium ions as their cofactors. Polymerases are of four types: DNA polymerase, RNA polymerase, T4, T7 polymerase, teeth polymerase. Again, DNA polymerases are of four types, which are prokaryotic: polymerase 1, polymerase 2, polymerase 3, polymerase 4, polymerase 5. And there are 15 known eukaryotic DNA polymerases. There are Pol A1, Pol A2, Pol B, Pol G, Pol G2, Pol D1, Pol D2, Pol E, Pol E2, Pol E3, Pol H, Pol K. These two pictures shows us the DNA synthesis using DNA polymerase and RNA polymerase. First pic shows there is one double-stranded DNA which on replication forms two strands, leading strand and logging strand. Using DNA polymerase, it converts into two double-stranded DNA molecules. And in second diagram, there is a single-stranded DNA, and using RNA polymerase, it converts into single-stranded mRNA. Ligases. It is a special type of enzyme that in the cell repairs single-stranded DNA molecule in simple word strands that have double strand break. Purified DNA ligase is used in gene cloning to join DNA molecules together. There are four types of ligases: DNA ligase 1, DNA ligase 2, DNA ligase 3, and DNA ligase 4. DNA ligase 1 ligates the nascent DNA of the lagging strand after DNA polymerase 1 has removed the RNA primer from the Okazaki fragments. Lagging strand is formed at the time of replication of DNA. First, it keeps RNA primers. After that, lagging strand forms Okazaki fragments. DNA ligase 2 alternatively spliced from the DNA ligase 3 found in non-dividing cells. DNA ligase 3 complexes with DNA repair protein. DNA ligase 4. It catalyzes the final step in the non-homologous and joining DNA double-strand break repair pathway. Transferases. These enzymes add nucleotide towards 3' end of DNA strand. They are also called as terminal deoxytransferases. We can modify cohesive ends to blunt ends. We can add any nucleotide of our choice like adenine, thymine, guanine, and cytosine. DNases. These enzymes degrade or destroy DNA. DNases are of three types: DNase 1, DNase 2, aminases. DNases are used to degrade DNA, and these enzymes perform random degradation because they are not specific to recognition sites. This two diagram shows the synthesis of DNA into fragments with the help of DNase1. And the second picture, DNA is digested with DNase enzyme. And second picture shows us that DNA is digested with DNase enzyme, and fragments are visualized in the form of polygraph. RNases. These enzymes degrade or destroy RNA. RNases are of two types: endoRNase 1, endoRNase, and exoRNase. EndoRNAs are divided into two types: RNase A and RNase H. ExoRNAs are also divided into two types: RNase 1 and RNase 2. Kinases and phosphatases. Kinases are the enzymes which add phosphate group at 5' end. It means that kinases adds phosphate groups at 5' end but not at the 3' end. Kinases are also, we can also call kinases as polynucleotide kinases. Phosphatases are the enzymes which remove kinases at phosphatases 5' end, whereas phosphatases removes phosphate group at 5' end. Phosphatases are also called as alkaline phosphatases. There are two pictures. First picture shows us the mechanism of phosphatases, and second picture shows the mechanism of kinases. In the first picture, phosphate removes the phosphate group, and in second picture, kinases add first picture phosphate remove phosphate group from DNA or protein, and second picture shows kinases add phosphate group to DNA or protein. Restriction endonucleases. These are also called as molecular scissors or restricases. Molecular scissors cuts the specific part of DNA which we need to clone. These comes under the family of nucleases. These are discovered in bacteria in the year of 1962. These enzymes are specific towards double-stranded DNA. They cut double-stranded DNA at or near to specific recognition sites called as restriction sites. Restriction endonucleases are of three types: type 1 restriction endonuclease, type 2 restriction endonuclease, and type 3 restriction endonucleases. All restriction endonucleases follow two rules. For circular DNA equal to N rule, and for linear DNA that is N+1 rule. While coming to applications of gene cloning, production of protein from cloned genes, it means it produces protein from cloned genes. Hence, production of recombinant pharmaceuticals in medicine. These recombinant pharmaceuticals have many benefits, and it is very helpful, and it does not have any side effects. Synthesis of human growth hormones in E. coli, and recombinant vaccines. It can, from cloned genes, it can prepare recombinant vaccines like vaccine for hepatitis B and various other diseases. So gene cloning is very important. Thank you for viewing my PPT. Thank you for hope you'll subscribe to my channel, and you may get inspired from this. Thank you.